Hello, everyone. Good evening, and welcome to yet another edition of the Indian Express Explained Live, earlier known as E-Explained, our series of explanatory conversations with domain experts, gurus, authorities on specific subjects. I am Monojit Majumdar, editor for explanatory journalism at the Indian Express. Explained is a unique project. It is unique because it is rooted in the news, but it goes beyond just the news. It is context, it is background, it is connecting the dots, it is explanation. It is what gives you a richer, fuller, more complete understanding of what is going on in the world around us. Today, we are talking about vaccines, specifically the vaccine that we all hope will defeat the COVID-19 pandemic and allow us to return to a degree of normality. What does it take to produce a vaccine? How is the research progressing? How soon can you and I get a shot that will protect us? And what will it cost? To talk about a lot of these issues, we have with us this evening one of India's most eminent medical scientists, Dr. Gagandeep Kang. Professor Kang, welcome to the Indian Express.
Thank you, Monojit.
We are honored to have you with us. Thank you for your time, ma'am. Speaking with Professor Kang will be my colleague and science editor of the paper, Amitabh Sinha. Amitabh will start with a short presentation, which will essentially lay the ground for the discussion that will follow. Thereafter, Dr. Kang and he will have an explanatory conversation for about 45 minutes. And then we will open the floor for a limited number of questions from the audience. Thank you for joining us and welcome once again. Amitabh.
Good evening, everyone. Good evening, Professor Kang, and thank you so much for joining us, taking the time out for this event. I'll just begin with a short presentation, and by short, I mean a short presentation, just to give a sort of lay of the land to people who have joined us. Most of them are already aware of the situation, but just so that everyone is on the same plane. So I hope you would be able to see this. 1 second. So just before we go ahead, so as most of you are already aware, there are about 169 candidate vaccines that are currently under development. There are probably more, but these 169 are the ones that are currently listed by the World Health Organization. 30 of them are in clinical trials, which means they are being tested on human beings. 139 of them are in preclinical trials, still being tested on animals. There are 2 Indian candidate vaccines currently in clinical trials. You would have heard of them. One of them is the Zydus Cadila, the other Bharat Biotech. What probably you haven't heard much about are that at least 8 other Indian vaccine candidates are in preclinical stages right now. Now, of course, you would have heard about the Russian vaccine, which has already been approved. There is another vaccine that has been approved in China, but that is approved only for limited use as of now. It's meant to be used only on Chinese soldiers. As of now, we do not know how many people have been administered that vaccine. So these are the leading candidates, the names that probably you are hearing about a lot these days. AstraZeneca in collaboration with Oxford University, that this is the vaccine that is the most in demand. Almost like 300 or 3 billion doses of this vaccine probably has already been pre-booked by several countries. This is also the vaccine whose human trials are going to start in India for the Indian market, for the Indian population. Then you have vaccine candidates from Moderna, Janssen, which is essentially the pharma unit of Johnson & Johnson, Pfizer, Sanofi, GSK. All these are very big pharma companies, very well-known brands. One, a little-known brand is Novavax. It's a US-based biotech company, but this vaccine is, a lot of people are betting on this vaccine as well. US has pre-booked several 100 million doses of this vaccine. Again, This is another vaccine which is coming to India. We already have an agreement. Pune Serum Institute has an agreement to manufacture this vaccine. So probably you will keep hearing about Novavax more in future. A little about the different stages in vaccine trials. Again, this is something that is being talked about a lot. Once a vaccine is developed, a candidate is developed, then it has to go through various stages of development. First, of course, is the preclinical trials, wherein it's tested on suitable animals. There are several rounds of animal testing that happens before it's found suitable to go to clinical trials and be tested on human beings. On the human trials, there are three phases. Again, something that's been discussed a lot. Phase one, usually on a very small group, a few dozen people are selected for these trials. It's mainly the safety of the vaccine is tested in this phase, wherein it's seen whether this does not produce any side effects in the human beings, whether it's like good for it, it's safe to be injected. Phase two is mainly about, so a larger group of about several hundred people are tested here. And then you have, the main point is whether it generates a sufficient amount of immune response in the human beings to be able to fight the disease. Phase three is the most large scale human testing. It's done in real life situations, not in laboratory settings. Usually several thousand volunteers are injected with the vaccine, and usually they are divided into two groups. One is given the vaccine, actual vaccine. The other group is given something which is not the vaccine, something else. And then they are allowed to continue their normal life, and they are evaluated after some time to see whether people who had been given the vaccine have they were able to better fight the disease. So that's the phase three. And after, at each of these stages can take, in normal circumstances, can take several months. After each stage, data is generated, that data is evaluated. In normal circumstances, it's also published, peer reviewed, and you go back to the regulatory authority for approval for the next stage. Now, in the current pandemic, these lines are getting blurred. Phase one and phase two testing is approved simultaneously. Phase two and phase three trials are being approved simultaneously. So these are, in the usual circumstances, you would find a lot of gap as well. So, and the last slide. So this is also something you might be hearing about a lot. Different types of vaccines. Some of them are DNA-based, RNA-based vaccines, protein-based vaccines. These are essentially different approaches to generate immune response within the human beings. How does a vaccine basically work? The vaccine just goes and tells your body. It helps your body's immune system to identify an enemy. Basically, in this case, a disease in the form of a virus or a bacteria. And when it tells you what the enemy is, how do you identify that? How does the immune system identify the vaccine? You either give the entire virus. So you inject a live virus or an inactivated virus, and you let the human immune response identify it and say, if you find anything similar to this, you need to resist. You do not allow it to enter your body. So live virus or inactivated virus would be similar to presenting a photograph of the picture and telling the person who to identify. The others would be, the other way to identify the virus or the enemy in this case is to know specific characteristics, the unique characteristics, the unique identifiers. And these are the different kinds of approaches, the different characteristics that are identified that can uniquely identify that virus. So these are used by the vaccine. It's injected inside your body so that the virus or the bacteria can be uniquely identified by the human immune system, and then it can generate the required immune response. So this is all about the virus. This is the last thing before we go on to the before we get our guest in for the day. Just 1 second. Okay. Professor Kang, I hope I'm audible to you.
Yes, you are, Amitabh. Thank you for that nice overview of vaccines.
Thank you. Before no, I don't want to delay further. So let's just get on with the first question. There seems to be a near unanimous view right now amongst experts as well, that a vaccine for novel coronavirus is near certain to be developed by early 2021. Do you share that kind of optimism? I mean, if we forget the Russian vaccine for the time being, which is already there, I am sure we'll return to Russian vaccine during the course of discussion. But apart from the Russian vaccine, do you share that optimism that we'll have a vaccine by 2021, early 2021?
I think it's highly likely, but it's not guaranteed. Until you actually have efficacy data on a vaccine, it is impossible to say whether a vaccine will work in humans or not. We have very encouraging data on immunogenicity. We have encouraging data from some studies in primate models that seem to indicate that some of the vaccines that have been evaluated in those systems do prevent severe disease. So that's all very good, but we have been led astray by animal studies before. So until we have data from humans, I would not be taking bets on it.
Right. When the outbreak had started in January in other countries and in March in India, had you thought that a vaccine probably would be developed so early, within a year, that it would be, and now that Russian vaccine is with us within just a few months, does that surprise you?
It does. It's incredible. I mean, to have 150 vaccine candidates all being developed for a novel agent, this has never happened before. If we look at the kinds of fields that have had multiple vaccine candidates before, maybe malaria, but malaria is a disease we've known for hundreds of years, and we've tried to develop many vaccines for the many stages of malaria. We have never had any single organism with addressed so quickly. I think some of this is the learnings from Ebola. In the case of Ebola, we had a vaccine that was sitting in a freezer and had not been taken further. And it took us almost 9 months from deciding that a vaccine needed to be tried out for Ebola to actually getting the vaccine into humans. So following that, the vaccine community and the global health community came together and said, this should not happen again. So there were a lot of discussions about what needed to be done to ensure that the next time an outbreak happened, we would be better prepared for it. Some of that thinking led to the formation of the Coalition for Epidemic Preparedness Innovations, which has multiple partners. But individual governments also thought about it a lot. Many of the programs that have been established that are doing a large part of the funding, for example, BARDA, which was established after anthrax, have really been in response to the need for handling infectious disease outbreaks. That said, I don't think anyone expected this to be quite this big. Certainly not for a coronavirus. The coronaviruses that we have seen previously, like SARS and MERS, were much more limited in their extent of spread. When we predicted pandemics, we thought it was going to be a new influenza. And now we know that it's not just influenza viruses that can result in this kind of devastation.
Right. Let's now get on to some of the queries that are probably uppermost in the minds of our audience here, also people outside. One, of course, is how soon a vaccine, when developed, is likely to reach us. Now, supposing the Russian vaccine is already being developed, how long will it take to reach us? What are the processes in between? What are the logistics? Also, if you can address the issue of affordability of vaccines.
I don't think I would call the Russian vaccine a vaccine that is ready for prime time. It's a vaccine that first went into humans on the 17th of June. It's been through accelerated phase 1 and phase 2 testing, which means that in a very small number of people, we know that the vaccine is not causing severe reactions and it is immunogenic. So just the fact that the vaccine has been licensed by one regulator does not tell us that this vaccine will work to prevent disease. We need phase 3 efficacy trials in order to do that. That's true for the vaccine that is licensed in China as well. Without efficacy data, I wouldn't think about having that vaccine. Now, in terms of how quickly will we get the vaccines, if we find that the Oxford vaccine works, and that is in phase 3 trials in multiple parts of the world, given that Serum Institute of India has already started to make the vaccine, I think that would be the first one that we would see where we would have data that would allow us to confidently think about using that vaccine. In terms of pricing, there's a range of prices out there. When vaccines first start out, and if they are made by multinational manufacturers, the cost of those vaccines is in the several hundreds of dollars. And for India, we know that if a vaccine is more than $3 a dose, we are going to be unable to take it. So the price that has been quoted for the serum vaccine in the order of 900 to 1,000 rupees a dose might be something that we could think about. And that, of course, is a starting price. All vaccines start out expensive. And then once you start to make lots and lots of the vaccine, you can bring prices down quite a lot.
Right. And what about the logistics, manufacturing, supply, accessibility? I mean, how much time does that take to arrange for these logistics to take shape so that it's accessible to everyone?
So this is the really complicated piece of the puzzle. I think people think that if we find that we have a successful vaccine, it will be at your friendly neighborhood doctor tomorrow.
Yes.
It will not. It takes time to make a vaccine, and the reason why vaccine manufacturers are now trying to buy facilities, book time at manufacturing facilities, is because they want to be able to make the vaccine even before it is proven to be safe and effective. So this is called at-risk manufacturing, and this is what Serum Institute has done. And this is what a lot of companies around the world are doing. They're making the doses of vaccine sufficient to get it out the door as soon as the vaccine is licensed after efficacy data are obtained. Now, that is the fastest way to get to the market. But the whole process of regulation takes time. It will take time to review the dossiers. It will take time to get the vaccine licensed. And once the vaccine is licensed, we have to think about the logistics of distribution. For example, some of the vaccine candidates have cold storage requirements. If you need to store a vaccine at -70 or -80, there are many countries where you cannot think of using that vaccine at all. So you need something that fits in with your cold chain infrastructure. You need to be able to buy the vaccine, you need to land the vaccine, you need to distribute the vaccine, you need to maintain it in a cold store, you need to train staff in how to give the vaccine. So the whole communications piece, including motivating communities and arranging vaccination sessions, is going to be challenging. One very important thing to remember is that we have never vaccinated all age groups before. Our vaccination program has mainly consisted of vaccinating children. And if you are pregnant, then in the antenatal period, you will get 2 shots of the tetanus vaccine. Other than that, we really have not in the public health program given out vaccines to adolescents, adults, and certainly not to the elderly. Now, trying to figure out how our immunization system, which is geared towards maternal and child health sessions, will now reach people at all ages and stages of life is a challenge. And then if we think about the fact that when you have the vaccine available, the vaccine is not going to be available in one tranche so that you can vaccinate everybody. You're going to have to think about prioritizing which populations need it first. Right now, the thinking is that it will be healthcare workers that will be prioritized, then the elderly, and then you need to decide how you define elderly. Is India the same as Norway, for example? And then people with comorbidities. So you also have to think about when you have a vaccination program, why are you vaccinating people? If you're vaccinating people for prevention of morbidity and mortality, you will go by risk groups. If you're vaccinating to prevent the spread of disease, then you should be vaccinating the young and healthy who are more likely to spread disease. So it's important to understand the purpose of a vaccination program when you decide how you're going to implement it.
Yeah, that's very useful, actually. The other question that is, that a lot of people are asking is how effective this vaccine is likely to be. And what we mean is probably the vaccines that are still in the pipeline and they're likely to come in the early part of 2021, especially when we know that the virus probably is still in a phase that it can mutate several times right now. So how effective any single vaccine is likely to be. Also the fact that, we seem to be amenable to accept a vaccine which is of low effectiveness. The US drug controller has already said even a 50% effective vaccine would be something that would be acceptable. Now, do we have examples of vaccines which are only 40% or 50% effective? And would not that level of effectiveness, would not that give us sort of false sense of assurance amongst people that they are immune? So all about effectiveness in that sense.
Yeah. So I think the first thing is the vaccine going to work, and in what proportion of people, and for how long will that protection last? Now, the US FDA and the WHO have said that a point estimate of 50%, which means you do a clinical trial, and in the clinical trial, you should have at least 50% of disease prevented through the vaccine.
Right.
That actually means, from the size of the US trials, that the lower bound of efficacy may be as low as 30%. And that is still acceptable to the regulator. So remember that these are first generation vaccines, and we are looking at a vaccine that prevents a reasonable proportion of disease. And that reasonable proportion is about half of disease. Now, how long will this vaccine give us protection? We have other examples of vaccines where the protection does not last very long. Influenza vaccines are a case in point. And going by the duration of antibodies to seasonal coronaviruses, it appears likely that we may find that the protection might be short-lived. Will it be months? Will it be years? We don't know yet. We'll have to wait for the results of the vaccine trials to understand what the initial levels of protection are and for how long that protection is maintained. The current follow-up that is planned in many of the trials is only 1 year. And within 1 year, it's unlikely that you're going to have a large number of reinfections, but it's certainly something that we will need to track. You brought up this issue of false assurance that you will feel like you have an armor and are protected if you have been vaccinated.
Right.
And that is a problem. I think if we had a correlate of protection, if we knew that among the people who are vaccinated, if you had a signal that you could describe, antibodies, for example, and you knew that those antibodies correlated with protection, then you'd be a lot more confident in saying how well the vaccines are working. Until we have that kind of information, I think even after vaccination, we'll have to proceed with caution and warn people that even though they have been vaccinated, protection may be incomplete. Now, you mentioned that, this seems like a low bar, but the malaria vaccine has 30% efficacy, and that has been licensed and under an emergency use initially, and is being tried out in countries in Africa. And the reason that the African countries want that vaccine is because even prevention of a small proportion of malaria still has huge health and economic benefits for them. If we take a look at cholera vaccines or we look at rotavirus vaccines in our children, the vaccine efficacy in the clinical trial that we did were 55 and 40%. Nonetheless, and a vaccine trial is an artificial setting. We are using those vaccines, and the vaccines prevent a significant proportion of severe disease. So a vaccine doesn't need to have 100% efficacy, but we do need to understand the parameters with which we are working.
Right. So a 40% or 50% efficacy vaccine would be good enough. It would certainly be better than having no vaccine at all.
That's right. And remember, these are first generation vaccines. If we understand how they work, then we can look at improving their performance. We can add things like adjuvants to them, see if that increases the proportion of people who are protected or the duration of protection. And given that in this situation, we are going to have lots of different vaccine candidates, we could very interestingly also think about the possibility of mixing vaccines. So
Right.
if you had more than one mechanism by which vaccines induced an immune response, then you might get a more durable response. So these are called prime boost strategies. And we could be thinking about that as well.
Right. The other thing which is again being talked about a lot is there seems to be, because probably with the speed at which the development is taking place, there seems to be a little bit of trust issues with these vaccines. Now, we don't know much about, I mean, in India, people are still very eagerly talking about it, but there have been some polls that are done in US and Europe where a significantly higher proportion of people seem to be extremely skeptical of the vaccine, and they are saying that they probably would not be the first ones to take the shots when they are ready. And one of the main reasons for this seems to be the speed with which these vaccines have been developed, and also the at least in part some of the trials have been bypassed. Both the Russian vaccine and the Chinese vaccine, which have been licensed till now, both of them have bypassed phase 3 trials. So how important is phase 3? What can go wrong if the phase 3 trials do not take place as designed?
So, you want people to be able to trust the healthcare system. You want people to believe that doctors and scientists and researchers are doing their best to make sure that we get high quality, safe, and effective preventive strategies out there. Anytime you get people that shortcut the system, that don't follow process, that place undue risk, it becomes a problem. So if we have the Chinese and the Russian vaccines are examples of where the system has been short-circuited in a way. And what that leads to is people being unsure whether these are really quality, effective products or not. I would not advise that. One way or the other, you need to prove that a product works before you use it. Now, there are regulatory mechanisms that allow you to do these emergency use authorizations, but then the requirement becomes a very high bar of following up everybody who receives the vaccine to ensure that at least after licensure, if it's an accelerated licensure, you actually treat the vaccinated people like they are in a phase 3 efficacy trial. You still have to follow them up. You have to make sure that they are safe, that nothing goes wrong. If we do things that way, then I think addressing one problem quickly and damaging the entire system is not a good way to go. We should be thinking about not just COVID, but what this means for future drugs and vaccines as well. The question that you brought up of vaccine hesitancy and the US surveys that say that a lot of people will not take vaccines. I think what has happened in the US is that even before this, there was a lot of doubts about vaccines. Some of this is around messaging, around the fact that we are constantly being told that foreign antigens are something that is dangerous, that you shouldn't be exposing your children to them, that you should not have combinations of certain kinds of vaccines because it might be dangerous for your child. Now, obviously, you don't want danger. Vaccines are given to healthy people. They are not given to people who are sick. So you don't want something that has side effects. You don't want something that is likely to have long-term consequences. So that is understandable. But the messaging that has been created around existing vaccines is just wrong. It has been disproved time and time again that the vaccines that are currently in the program are safe, can be given together, do not lead to short or long-term side effects. Unfortunately, when you have celebrities, when you have people who have a social media following, who are good influencers and persuaders, who constantly repeat this conspiracy theory that vaccines are a tool being used by unknown forces to damage all of us, then that becomes something that a certain section of the population does begin to believe. And perhaps the fault lies with us as a medical community that we don't do enough to counter this with credible voices. So America is in a situation where a lot of people don't think that vaccines are going to be something that they will sign up for. But on the other hand, also in America, you have a group that is called One Day Sooner, which is volunteers who are signing up for human challenge studies, saying that if you want to evaluate a vaccine for COVID-19, I am willing to take the vaccine and then be challenged with live virus so that you get efficacy data quicker than you would otherwise. So you have people with a very firm belief in science, and you have people who have no belief in what the scientific system offers.
Professor Kang, there are suggestions that have been made and are repeatedly being made that while we await for a vaccine to emerge, in the meanwhile, in the interim, why not eat so and such thing to boost your immunity, or do such and such thing to boost your immunity? Can immunity be boosted?
Well, there are certain things that we know about that affect your immune responses. An example of that is zinc, which plays an important role in infections. Iron plays an important role in bacterial infections. There are studies that have looked at vitamin A, which are older studies, and then there are studies that have looked at vitamin D. Now, all of these seem to affect immunity in a situation of deficiency. When you don't have enough of that, it might affect some aspects of immune function. But it's important to remember that unless you are severely malnourished, you're not likely to be in a situation where you have a deficiency that will have a functional consequence when you encounter a pathogen. So immunity boosters would be wonderful in principle. In practice, I don't really know of one.
Right. Uh, what we are also seeing is a lot of these vaccines that are being developed, their human trials are being conducted not in the countries where the development is happening, but in mostly third world countries, Brazil, South Africa, increasingly now in the countries of the Middle East, Saudi Arabia, Kuwait. Uh, why is that happening? Why are we seeing this phenomenon?
So I think one of the things that we have to remember is that phase 3 efficacy trials require disease.
Right.
Right? You need to have a reasonable expectation that there will be sufficient numbers of cases for you to be able to measure the effectiveness of a vaccine. So when you design a study, you design it predicting how many people are likely to be infected. So the current size of the US trials, for example, is 30,000 individuals. And the idea is that in those 30,000 individuals, in a relatively short time frame of several months, we will see at least 150 cases.
Right.
If a vaccine is going to be 50% efficacious, 100 of those cases will happen in the 15,000 who got placebo. Right. And 50 of those cases will happen in the 15,000 who got vaccine. So you will have a vaccine that is 50% efficacious. Right. But you need to have those 150 cases. So if you look at China, China is leading vaccine manufacture. They've got all kinds of products that they are evaluating. And they can do the phase 1 and phase 2 studies, but they don't have disease. So how are they going to do phase 3? Which is why the Chinese are in the UAE, in Saudi Arabia, in Brazil, with all of their vaccine candidates, because these are places where there is a reasonable expectation that vaccinated people will be exposed to infection. So it's not about treating third world countries as guinea pigs. It's just about third world countries. And well, then the US is a third world country because they have the Moderna trial is being done at 89 sites in the US.
Right. But there is a new term that has emerged in this entire vaccine development story, and that is vaccine nationalism. You know, countries rushing forward to sign advance agreements with companies to secure supplies of vaccines that would be produced, may not be produced, but then they are hedging their bets, and several million doses, hundreds of millions of doses have already been secured. Now, potentially, this is being done only by countries which have enough dollars to sign these initial agreements, and these are worth billions of dollars. And we have seen how the United States has already reserved about 800 million doses. The UK has done about 350 million doses of vaccine reserved. So that can potentially, make the vaccine, at least in the short term, inaccessible to large areas of the world who did not have the foresight or the money or the resources to make advance agreements, isn't it? How do you see this development?
I think vaccine nationalism, or the question of access, is one that has been discussed almost from the beginning when the vaccine programs began development. And the reason for that is the world has seen this before. We saw it with H1N1 when WHO tried very hard to establish a global allocation mechanism, but it got such few doses of vaccines from the vaccine manufacturers that it almost wasn't worth their while to invest a lot of time and effort in distributing those. Now we have something that is called the ACT Accelerator, which is the Access to COVID-19 Tools Accelerator, which has multiple partners, but the idea there is access not just to vaccine, but also to diagnostics and drugs.
Right.
And WHO is responsible for coming up with a fair allocation framework for deciding which countries, regions will get access to diagnostics, drugs, and vaccines through various implementation partners. The implementation partner for delivering vaccines, for example, is the Gavi Alliance. Now, will multinationals, will the successful makers of vaccine provide vaccine to the global allocation mechanism? These are the negotiations that are happening now. And the idea is from the global pool, there will be distribution of vaccine around the world. Now, the counter to that, which is we are all looking after ourselves first and not thinking about global good, is something that we are seeing. Governments have a responsibility to their citizens. And in the US, they've taken it to the extent of saying that, we want to buy a company that might potentially have a product we are interested in. We will fund you to do your trials. In some ways, what you would like is for everybody to be a good global citizen and share resources so that people around the world who need them the most get them quickly. But that clearly is not happening. We need to do our best, but then the way to think about it is there aren't 800 million people in the US. So if we do wind up with the US having 800 million doses of successful vaccines, they're not going to be able to use it all. So I'm assuming that they will give it away. And at least some countries in the region, for strategic reasons or whatever, will get access to those products. It's a worry. And it's something that we need to be thinking about. We have one of the largest populations in the world. There is no way that the Indian population is all going to be able to get vaccine quickly. So we need to be thinking about our populations and coming up with a clearly communicated strategy about how we will vaccinate and whom we will vaccinate and when. And I believe that the committee that is chaired by Dr. Vinod Paul is doing precisely that.
Right, because as you mentioned, the ACT Accelerator program, I mean, WHO has gone on record saying that there isn't enough money coming into the platform, the COVAX platform, to accelerate the development and deployment of vaccines. So they have already said that they require billions of dollars and very few money is flowing into that. The countries are actually wanting to sign independent standalone agreements. So they find that a more secure way of assured supplies.
So what countries are doing in many countries, for manufacturing facilities, for examples, countries had a step-in clause saying that we will build this manufacturing facility for you, but if there is a time of national need, we will step in and take over the time at the manufacturing facilities. This has happened in the US. Countries also have arrangements with vaccine manufacturers that are in their country, saying that if there is a biosecurity need, you are obliged to give us a proportion of what you produce. And that may be the entire supply, it may be 50%, it may be 30%, it really depends on the agreements with the country. So many manufacturers are constrained because they don't have manufacturing facilities of their own, or because they have these prior agreements with their governments where they have committed to supply a proportion of their manufacture to the government. So to counter that, there are arrangements that both Gavi and CEPI are doing, which are to promote access. So Gavi is doing advanced purchase commitments, talking to manufacturers to make sure that when vaccines are available, these vaccines will be bought by Gavi. It's already done that with multiple companies. And CEPI, no amount of money is enough when you have 150 candidates to support. But the programs that CEPI is supporting, many of them have now been taken over by other funding agencies. The Moderna example is a prime one. Right. First one to be funded, first one to go into human clinical trials, now being paid for almost entirely by the US government. But the first tranche of funding that they got was from CEPI. So everybody's trying to do their bit. At some point, maybe in the next pandemic, we will figure out which are the platforms to focus on. Because before this, we did not know how RNA platforms, how DNA platforms would work. We did not know how the viral vectored platforms, of which we are seeing many, would work. So in this pandemic, we have a mix of traditional technologies, the subunits, the inactivated vaccines, as well as very, very novel technologies that have never been licensed before. Maybe the biggest lesson that we will learn from this pandemic is that you will not need 150 candidates the next time around. You could potentially do it with a high probability of success with a dozen.
The last two questions before we open it up for the audience. One is now the Serum Institute would be conducting human trials for the AstraZeneca Oxford University vaccine in India. What is the process for use on Indian population? Does every candidate vaccine have to undergo a human trial, phase 3 trials in India as well before it can be made available to Indian population, or we can go ahead and straight away, make it available to Indian population as well?
When you have a product that is licensed in a different country and that country has what is known as a stringent regulatory authority, then getting a vaccine licensed in India requires a very small trial in India. These are called bridging studies. You can do it in less than 100 participants. And if the immunogenicity data from that study is similar to what was done in the place where the vaccine was licensed, then the vaccine can be accepted in India. When the vaccine is a novel product that has not been licensed elsewhere, then you will need larger scale studies. Given that the ChAdOx1 vaccine is in phase 3 studies that are much further advanced in other parts of the world, it's likely that by the time the studies are done in India, there may be phase 3 efficacy data, and the data that Serum generates now on their 1,600 participants can be used for submission for Indian licensure.
Right. The last thing, this entire process of developing a vaccine for novel coronavirus, the way it has been done, so many candidate vaccines coming in, so much of urgency being shown, huge amount of funding being received. What are the implications for future vaccine developments? I mean, what is going to change for the other vaccines that we are developing for other diseases after this experience with novel coronavirus vaccine?
I think for many of the technologies that we have for vaccine development, right? Many of the new vaccines that we are developing are actually based on pretty old technologies. So in the 1950s, we got cell culture that allowed us to make polio vaccine, rotavirus vaccine, a number of vaccines. In the 1980s, we got conjugation technologies that allowed us to do pneumococcal and Hib vaccines. After that, in 2010, when we got DNA sequences and then could look at this as a good target for making a vaccine, that was the first time we started to use new technologies, reverse vaccinology approaches to make vaccines. Since then, in terms of licensing vaccines, the new technology has really been the Ebola vaccine, where we had the first vectored vaccine, viral vectored vaccine being licensed. We have not so far had an RNA vaccine or a DNA vaccine licensed. We have not had new adjuvants available. We have not tried prime boost strategies. So if these are all learnings from this pandemic, I think that is what is going to change the face of future vaccine development.
So in the different approaches that are being tried, the new technologies, at least in this pandemic, is there any difference in the probabilities of which particular approach can deliver a successful vaccine? Just in terms of probabilities?
I think, okay, here is where we can take the wild bets. But if you were to ask me, I think we are more likely to have a successful mRNA vaccine than a DNA vaccine.
Okay.
But we can see next year how we do with that. But I think more than, if we look at vaccines, one of the things that we have to think about is scale.
Right.
And so far, the traditional technologies have allowed us to make hundreds of millions of doses. The new technologies, because they are more complicated approaches, could only scale to the low millions of doses. Now, vaccine companies are talking about 100 million doses of an mRNA vaccine. But we don't know the production issues with that. We don't know what manufacturing can mean in terms of quality of different batches for these kinds of vaccines. With the older technologies, we know this well because we've had multiple candidates use those technologies. So there's a lot to learn, not just about the vaccine technologies, but also about the manufacturing.
Right. We'll move on to some...
Amitabh, just before you move on, there's just one thing that I'd like to press Dr. Kang on. This is something more basic. This is what you actually started by asking. This extraordinary speed with which this has been developing. I mean, the fastest vaccine that the world had before this was what? The mumps vaccine, 4 years or so. Now, when Operation Warp Speed was announced in June, they said 300 million doses by January 2021. And it's been less than a year, and we have reached so far that you started by saying that you would reasonably expect this to happen. What is it that, where are we taking the time off? I mean, what is it that we are not doing which is allowing us to progress at this speed?
Well, one thing is that when there isn't a pandemic, regulators take their time and processes take a while. So there are, in our case, we have had regulatory windows only since the last year and a half. But even the FDA, which had windows, took its time over reviewing dossiers that were presented at different stages. Most of the shortcuts have been taken in, one, how much the window is for review of documents and obtaining permissions for further studies. And second is in doing parallel studies. So can we go into humans without going into animals? Can we go into humans and animals at the same time? How do we decide what is appropriate? Can phase 1 and 2 be combined? Can phase 2 and 3 be combined? Can we give permission for phase 2 at the same time as giving permission for phase 1? So not about shortening the rigor of the readouts from the trial, but just making sure that the process is as facilitatory as possible.
Right. So we move on to some audience questions, and I think our first question is coming from Mr. Vikas Singh, who I believe is joining us from Bokaro Steel City.
Yeah, good evening, sir.
Good evening.
Sir, good evening, ma'am.
Good evening, Vikas.
Ma'am, I am a layman and I will ask question like that only. So, ma'am, as per media report, the novel coronavirus is getting mutated, that we all know. And also I have heard that many countries has approached no lockdown, and they think that people will develop herd immunity with time. Also, somewhere I have read that there are few cases of vaccine-induced diseases, like a child developing polio after taking the polio vaccine. So, ma'am, considering these factors, as a common person, I have apprehension that is it wise, or is it really needed to take the novel coronavirus vaccine when it is commercially available? Your expert opinion, ma'am.
So if we look at prevention of disease, you can prevent disease happening in the future by being naturally infected. And for many things, that's what happens to us. You know that children get diarrhea a lot when they are young, and then when we become adults, mostly we get much less of that. Same for many kinds of other infections. Children are exposed to disease for the first time, they get sick with it, they develop immunity, and we go on. The problem comes that it's not always possible to predict who will develop severe disease and who will not, right? So if we look at measles, for example, we have a measles vaccine, but sometimes children who get measles have nothing. They have a fever for 2-3 days, a rash, it goes away and it's over. But measles can also kill children. Now, which child will be badly affected, which child will not, we don't know. And that's why we vaccinate. If there was a guarantee that everybody would have only mild disease, maybe we wouldn't bother so much. So in the case of coronavirus also, this is an unpredictable disease. In many people, it is asymptomatic. In many people, it's mildly symptomatic, but it also kills some people, and we don't understand what leads to the severe disease in some people. So that's why we think a vaccine is a good idea. Now, if you look at what has been happening to other states, other countries, in terms of lockdown and no lockdown, Sweden is an example of a country that decided it would not do lockdown. The UK did a lockdown, and yet mortality rates were similar across the two. Now, there are multiple factors that could explain that, but neither of those two countries reached anywhere near the levels of population immunity, or in terms of sero surveys, that were predicted to be required for herd immunity. However, we've seen in crowded places in Pune and Mumbai, we are actually much closer to the predicted number, which was somewhere around 60%. Pune is, in some places, is 50% plus, and again in Mumbai, in some places, 50% plus. Now, we have to remember that these are predictions. These are based on what modelers thought are likely to happen in the case of a disease that spreads in the way that the novel coronavirus does. Now, herd immunity has been shown is very important for preventing spread of infections. Will it kick in at 50%? Will it kick in at 60%? What does that mean? That is something that we need to think about at both the population level and at the individual level. So let's say that in locality X, right, you have 70% of people infected. So if you are in the 30% that is not infected, you have herd immunity because those 70% who have been infected are not transmitting disease anymore. They're not allowing it to spread. But if you come out of that locality and you move to another place where there is only 10% of people who have been previously exposed, now you are vulnerable again. You can be infected because in this community, the chances of spread are much higher. So relying on herd immunity instead of vaccination can be quite a dangerous concept if you do not, I mean, unless you don't want to move out of a place where you are protected. You brought up the question also of vaccine-induced polio. And this is something that we see in polio in about less than 1 in a million children who receive the polio vaccine. There are other side effects that are much commoner. Now, if you look at polio 50 years ago, children died of polio. When I went to school, in every class, you would have one child that was wearing calipers because they had been affected by polio. And remember, these were the people who survived with a disability, not the ones who died. So polio is not gone. And what we did was we made a choice. We said, in order to prevent death and disability due to polio, we will take the risk of 1 in a million children potentially being affected by polio. In every situation, whether it is a vaccine or a drug, it's very important to do this analysis of what is the risk and what is the potential benefit, and do it at an individual level and for the whole population. For example, when we take drugs, right? We take an antibiotic, we know we will get diarrhea, but we take the antibiotic anyway because it is going to cure us of something else. So the benefit is the cure, and the risk is that we will get a side effect. And when we are sick, we are willing to make that compromise. Vaccines are given to healthy people, so we expect a much higher safety margin for vaccines.
Professor Kang, since you mentioned the sero surveys, I'm just tempted to ask you about these sero surveys, the results that have just come in from Pune, Bombay, and Delhi earlier. How are these to be read? Should we feel apprehensive that the disease has spread amongst us so much beyond what we were thinking that it has? Or should we be happy that it has already reached a stage from where probably we can hope that the phenomenon of herd immunity starts to approach? So how should that be seen?
I think the one thing that is most clear to me is that we have a finding here of a high level of seroprevalence that's higher than anywhere else in the world. Now, why is that happening? And what does that mean? You can make a simplistic assumption that if you have a population that lives in as dense areas as ours, then spread is likely to be higher. How do we use this information? We could use it to say that in a population that has over 50% or 60% seropositivity, maybe you don't need a lockdown because they're not going to have too much spread anymore. You could use it to say that maybe these are the people who should be allowed out to work because they've already had such high levels of exposure that they would be, at least in the short term, resistant when they are re-exposed. They may not get reinfected. You could look at it by saying, wow, if the proportion of spread is so high, so much of our communities are being affected, then maybe all the numbers that we are calculating in terms of severe morbidity and mortality need to be revised. You know, right now we are going by cases that depend on RT-PCR or antigen tests. But the real infection rates are the infection rates that are shown to you by serosurveys. They really reflect what has happened in the population that may or may not have been sick. So we do need to use these data very carefully to help our planning for the future.
Right. Thank you. Our next question is coming from Mr. Tejas Missal, who is joining us from Pune, I believe.
Hello.
Yes, please.
Hello,
go ahead.
Tejas.
Hello. Am I audible?
Yes, please go ahead.
Thank you, sir. Thank you, ma'am. Before I put my question, I just want to say that it's been very informative session. My name is Tejas Misal. I'm student of MBBS. My question is, the Russian health officials have announced that they have got the regulatory approval for Sputnik 5, the vaccine they developed. Ma'am, how can a vaccine get a regulatory approval without going into phase 3 placebo-controlled clinical trials? Isn't it against the medical ethics of vaccine trials? And if not, then can India get such fast-track approvals for a vaccine so we can go ahead with our vaccine, with our own vaccine? Thank you, ma'am.
Tejas, thank you very much for that question. I think you raise a very important point. And countries have national regulatory authorities, and the national regulatory authorities function by their rules. There are provisions in the rules for accelerated approvals or for emergency use authorizations. So licensure can be given at an earlier stage than is usually done. However, when that licensure is given, then you need to make sure that the requirements of all drugs and vaccines, there are 3 attributes that regulators need to look at. Those are quality, safety, and efficacy. Quality is in the manufacture. Safety obviously comes from pharmacovigilance, and the efficacy comes from having a clinical readout. Because a regulator's responsibility to a product is through the entire life cycle of the product. It does not stop with licensure. So if a regulator gives an emergency use authorization, they have a responsibility to get post-licensure data. Then in this case, they will need to continue to collect data on safety and on efficacy for the vaccine that they have just licensed.
Thank you. Our next question is from Mr. R. Alfonso from Mumbai.
We can move to the next one.
Okay, I think Mr. Alfonso is not available. So our next question would come from Mr. Devashish Das from Cooch Behar in West Bengal. Yes, Mr. Das, go ahead.
I have one question.
There is an audio problem, Mr. Devashish Das. Sorry, we're not able to hear you. Can you fix your audio, please?
Yeah. I unmuted my set. Whether now it is audible?
I'm afraid you're not audible still.
There is a lot of disturbance, Mr. Das.
Yeah, there's a lot of disturbance. We'll come back to you, Mr. Jha. Just try to fix your audio, and we'll try to come back to you. In the meanwhile, the next question would be from Mr. Vidyanand Jha from Kolkata.
Just a second.
Is Mr.
So
Jha?
thank you. Yeah, thank you, Amitabh, and thank you, Dr. Kang, for a very lucid talk, actually. And it clarifies a lot of things. The question which I had in the beginning, that assuming that a number of actually these 150 vaccines are available, how should one go about choosing actually which vaccine to go for? Of course, it's a hypothetical question right now, but given the speed, with which, everybody aims to do that, maybe it will be a reality. I have some pointers from your earlier, questions and whatever you said, but it would be interesting if I hear from you on this.
Thank you, Mr. Jha. One of the most innovative things that has happened during this pandemic is the idea of doing multi-stage, multi-site comparisons of products. And we've been doing this in clinical trials for drugs. So we've all heard about the solidarity study and the recovery study and the discovery study that have evaluated various kinds of drugs in parallel arms to see how patients did. That has resulted in some drugs being dropped and other drugs continuing. A similar approach is being planned for vaccine trials. WHO is doing the solidarity studies, as phase 3 of solidarity will be vaccine trials. And they are currently recruiting sites to participate in these trials. They have 17 manufacturers that are interested in having their vaccines evaluated in this kind of parallel study. In addition to this, under Operation Warp Speed, there is a program called Active, where even though the intellectual property stays with the company, the vaccine trials are being done at sites that have been identified in partnership with the National Institutes of Health. And there is a common data monitoring committee and an independent statistical review committee that will be looking at the data that is generated from all of the candidates that are supported under Warp Speed. So this way you get a comparison of products at early stages of testing, because head-to-head comparisons of vaccines very rarely happens. Now that is for later testing, mostly in phase 3. In phase 1 and in phase 2 of testing, vaccines fail mostly because they are either not safe or not immunogenic enough. So they drop out pretty quickly. The most expensive stages of testing and the most time-consuming ones are phase 3 trials, which is why these innovations in comparison of vaccine products are something that will be valuable and will help us get vaccines quicker.
Thank you. Now we have a question from Mr. Anthony Kolanur, who is joining from Kochi.
Hello, I'm Dr. Anthony, a pediatrician and a ex-UNICEF health specialist. And I always get mesmerized by Madam's virology tutorials. And this evening also it was very useful. My question was, will there be uniform access and sharing of the newly developed vaccine at affordable cost to the countries? You had partially answered this question in the ACT Accelerator and global allocation framework for global pool and Gavi. But my specific question now is, assuming that AstraZeneca or Moderna will get the patent, being the first producers, will they ensure that they follow uniform availability and access, and it will be marketed affordably in all the countries by the stipulation of WHO or Gavi? If not, will the individual countries will be able to enforce the compulsory licensing clause of the Doha Agreement or TRIPS clause? I mean, that is with relation to the Patent Act. Will there be countries, individual countries will be able to work on that?
Right. Thank you. Yeah.
Thank you, Dr. Anthony. Now, when it comes to looking at access and affordability, obviously these are critical. And Gavi has taken on the responsibility for all of the 78 initially Gavi eligible countries, as well as taking on the responsibility for those countries who have signed up for the COVAX facility, which is going to be under the ACT Accelerator. So the doses that are available to the global allocation mechanism will be made available through that. And as with everything else that Gavi does, they will be affordable. Now, that is assuming, at least initially, that this is going to be a short-term approach. If it turns out that this vaccine is one where you need to keep giving repeated doses year after year, then I can see this becoming much more of a challenge. With regard to companies and when they will have their products licensed, I think there are a lot of discussions that are ongoing already from countries that have the capacity to manufacture vaccines, such as India, to be able to license the technologies. All of the major vaccine manufacturers in the world have made it clear that at whichever level, whether it is technology transfer, whether it is the ability to do fill finish, they are willing to make their facilities available, not just for their own products, but for any products that are licensed. And regulators have agreed to work with that. Compulsory licensing, in a time of global demand, it is very easy for a vaccine manufacturer to walk away from a country that insists on that. So I think looking at the mechanisms that are currently in place and in development, which are partnerships between industries, partnerships with governments and organizations under the ACT Accelerator, are the better way to go.
Right. Thank you. We now have Mr. Harshit Sharma from Ottawa. Oh.
Sir. Can you move to the next one?
Okay. The next question coming from Mr. Viresh Mahajani from Solapur. Mr. Mahajani, is he available?
Mr. Viresh, can you unmute yourself? Can you please unmute yourself?
Mahajani?
We'll come back to him, I think.
We'll come back to him. We just move on to Mr. Vinayak Joshi from Mumbai.
Yeah.
Products that are licensed by their national regulatory authorities, if they want to be able to sell their vaccine in other parts of the world, they can apply to WHO for something called pre-qualification. So many of our vaccine manufacturers are pre-qualified by WHO. This means that they have submitted a dossier to WHO, it has been reviewed, and if they are considered pre-qualified, then UNICEF can buy, any UN agency can buy products from them. So while WHO is not a regulatory authority itself, it does provide advice on regulations, and it does, for the purposes of pre-qualification, review products.
Right, thank you. Our next question comes from Chandan Nagar in West Bengal from Mr. Somadeep Chakrabarti. Is Mr. Chakrabarti available?
Can we move to the next?
Okay, so we'll come back to him. Next, we have Mr. Milan Mehta.
Yeah.
Am I audible, sir?
Yes, there is a lot of background noise.
One second, sir. 1 second. Only 1 second. Now am I audible clearly?
Please go ahead. Please go ahead.
Yeah. Good evening, and thanks for Indian Express for taking the hottest topic which is going on everybody's mind. My query to ma'am is that just 5-6 days back, there was a news that 73 variants have been identified of SARS-CoV-2. So how do you think it is going to affect the present vaccine development which is going on? And what if in future another variants are identified? That is one query. And second, based on what you gave a feedback, I have a different opinion regarding the best methodology for development of vaccine, which you preferred mRNA over DNA. I think spike protein may be the best way because that is only identifiable and the new mutated dangerous mutation developed of this virus. Thanks.
Thank you, Mr. Mehta, for those questions. In terms of the virus variants or mutations, this is an RNA virus and it has a very long genome, 30,000 base pairs. With RNA viruses, we know that when these viruses replicate, there are errors when the new RNA is being made. This coronavirus is peculiar because it also has a proofreading mechanism. So mutations in this virus are actually less than in many other viruses. Nonetheless, RNA viruses do mutate much more than DNA viruses, and that's what we are seeing here. Now, but changes the ability of the immune response to recognize the virus. That's when we absolutely will need to change the vaccine. That has not happened so far, and we hope it won't. Now, in terms of my saying mRNA vaccine, that's actually the mRNA vaccine that encodes for the spike protein. So one of the worries in the vaccine community is maybe we are focusing too much on the spike. We should be looking at other proteins of the virus as a backup strategy in case all of our spike candidates don't work.
Thank you.
Thank you, ma'am.
Thank you. We'll try to go back to Mr. Debashish Das from Cooch Behar, who previous time we were not able to hear him properly. Hope he has been able to fix the audio. Mr. Debashish Das.
I am audible now. Yeah.
Please go ahead. Go ahead.
Already when I have ended the question is that actually it's hard.
So that's there's a lot of disturbance. I'm sorry.
Now,
just try to keep distance from your mouth. Yeah, please go ahead. Let's try once again. Are you there?
I think he logged out by mistake. He must be joining back again. We take the next one.
Okay, so we go back to another one who we had missed out on, Mr. Alfonso from Mumbai.
Hello, am I audible?
Yes, please.
Yes,
sorry,
good evening.
Rachel Alfonso. Yes, please go ahead.
Good evening. Thank you for this informative updates on the pandemic. My question is, will India verify the efficacy of any vaccine that comes to India, especially considering the fast track nature of the studies, the racial differences, because many studies are done abroad, and the status of malnourishment in the Indian population. So will we verify these outside these vaccinations from other countries, considering the Indian status? That's my first part of the question. And secondly, could you address the issue of reinfections? Because if reinfections are happening in the absence of vaccines, which means the antibodies aren't working long enough, what is the point of a vaccine if reinfections are going to happen? Thank you.
So the first question is one that we worry about all the time. Will vaccines work in different populations? And certainly genetic makeup, malnutrition, prior exposure to infections can all modify how a vaccine works. The way it's usually done is that for the purposes of licensure, you probably would not want to repeat another whole efficacy study. You will do an immunogenicity study and license the vaccine if the immune response is good enough. So it predicts that the vaccine will work at least similar to the way that it has worked in a place where an efficacy trial has been done. But it would be very valuable to do what is called a phase 4 effectiveness study after the vaccine is brought into the program in India, and specifically addressing the question of populations that are different from other parts of the world would be very important. So testing the vaccine in malnourished people, testing the vaccine in the rural elderly, which may have a completely different prior exposure pattern. There would be lots of subpopulations where understanding effectiveness would be very important. Now, in terms of looking at reinfection, there are not a lot of documented cases of reinfection. One of the problems with defining what reinfection is requires us to have access to the first and the second strains of the virus and demonstrate that they are different. So just having a PCR positive at 2 points in time does not mean reinfection. Ideally, what you want to have is cultivable virus from both episodes of infection. As far as I'm aware, that is not really available for anyone. So we don't know whether reinfection really happens or not. Given what seasonal coronaviruses do, it is possible that reinfections will happen, but in what kind of time frame? If I am infected today, can I be reinfected next week? Very unlikely. Can I be reinfected 3 months from now? Again, unlikely. If I'm reinfected 1 year from now, is that feasible? It's possible. We don't know yet. We haven't had the virus for a year. Is it possible 5 years down the line? Absolutely. Happens quite often with respiratory infections. So what is it that we are looking to a vaccine to do? Does the vaccine have to do as well as natural infection, or should it do better than natural infection? The hope always is that a vaccine will do better than nature, that you will have one shot of the vaccine and you will be protected forever. But those kinds of ideal vaccines are very hard to make. So that's why right now what we are looking for is a vaccine that is 50% efficacious in preventing virologically confirmed disease.
Thank you, Professor Kang. I think we'll have to leave it here. Most of the questions that are coming with, I hope we have covered most of the questions that are coming in. A lot of them are variants of the same thing: how soon the vaccine is going to come to us, how much is it going to be priced, and also some of them have already been addressed. Unfortunately, we do not have time for all the questions that we can take. Thank you, Professor Kang, for joining us. Thanks once again. It's been very, very useful, and I'm sure the audience would have liked it also. So thank you everyone else for joining us. I hope that at least some of your questions were answered, if not all. Thank you.
Thank you. Thank you very much.
Thanks and good night.
Yeah,
Good night. Thank you.
Thank you.